Thanks for the invitation. It's good to be back. So here's the titles, and I started my research program at the Texas State about 1.5 years ago, and I belong to Department of Biology. So this is a fairly brand new research program, and this is one of significantly overused slides where plant immunity people try to explain how plant immune system works. Nonetheless, I'll try to use it today again. Here we have basal resistance. And the plant generally recognize the presence of pathogen by recognizing the PAMP, the pathogen-associated molecular pattern that include flagellin and other PAMP. And in this case, if the pathogen is around, then the plant has one receptor that names PAMP recognition receptor. So that includes FLS2, EFR. There's a lot of research going on to figure out how that signal goes into the inside of the cells. So once plant recognizes the pathogen, in this case flagellin is being recognized, and that will trigger several cascade of signaling events, and that will lead to activation of immune response, and collectively we call this one PAMP-triggered immune responses. Now, the pathogen we know long overcome this basal resistance, and we also know that the pathogen delivery is encoded protein inside of the cells. In this case, this pathogen utilizes type 3 secretion system, and the sole purpose of this effector is to suppress this basal resistance. So they'll essentially block this, the PTI, PAMP-triggered immune responses. Now that's where I will introduce resistant proteins, allergen-mediated resistance. That's also called effector-triggered immunity. In short, we call it also ETI. And what it does is that the allergen, the resistant gene that is from plant, they'll recognize the presence of effector, and they will trigger a series of responses in plants. And the hallmark of these effector-triggered immune responses, a lot of time this will accompany a program cell death known as hypersensitive response. So next slide shows how effective this allergen-mediated resistance can be. Here we have 3 potato cultivar that is infected with the Phytophthora infestans. That's very devastating oomycete pathogens. That is a causal agent for Irish famine. This cultivar is essentially highly similar except for one component, that is resistant genes. And you can clearly see after about a month later, the outcome is very contrasting. Okay. So this will drive all these plant breeders, plant pathologists to looking for how this allergen-mediated resistance operates. And that was a question that I had about 10 years ago. And by that time, we had a fairly few resistant protein already cloned. And that includes that resistance gene is from various plant species. And also, it actually has a very conventional, so lots of resistant protein belongs to many different classes. And it turns out several of them actually have very classical domain structures. In this case, in the middle, there is a nucleotide binding domain, and in the C terminus, there's a leucine-rich repeat domains. And this can be further subdivided into the 2 subgroups, depending on what is in the N-terminus. This is toll interleukin receptor and cold domain. So that's generally how we divide this resistant protein into, because a lot of resistant gene belongs to these particular groups. What is also known is they are found in many different plants, but also it is effective against many different pathogens as well. In this case, we have a bacterial cells. There's antibacterial resistance mediated by LPS2, LPM1. HLT, which I'm going to talk about very soon, that is against viral pathogen, TCV, Tony Kunkel virus. Also, it's even effective against nematode, insect, and fungus. So it is known that it's effective against many different types of pathogens. So we decided to study. And back then, the Dr. Classic lab has a very good plant patho system utilizing HLT resistant gene in Arabidopsis, and we were studying TCV, Turner-Kunkel virus. And that's arguably one of the simplest plant patho system available. So what we decided to do is let's do the genetic screening. And to facilitate genetic screening, we created a transgenic line. So DI17 is the ecotype of Arabidopsis that carry HLT resistant gene. HLT stands for hypersensitive response to Turner-Kunkel virus. Okay? And we put effector genes, TCV coat protein. We knew that what the HLT recognized, so we put that in under inducible promoter, because if not, then the expression of this CP will drive a hypersensitive response, and after that, the cell dies. So we put it under the inducible promoter. Upon inducer treatment, that CP will be induced and expressed, and there will be interaction between this, and that will drive cell death. So we are looking for a mutation that block this, because obviously the one that doesn't have impact on this particular process will die, and the one that we are interested in will survive. So that will be easy genetic screening. So here is actual genetic screening, and as he anticipated, vast majority of them when die, and there are several occasional survivors named CRT1. That's a compromised recognition of TCV-CP, and this is the one that I'm going to talk about today. Okay. First, we found that after all this map-based cloning, we realized that the mutation is in CRT1, and that belongs to GHKL ATPase superfamily. And we found that this CRT1 has all this conserved domain that is necessary for ATPase activity. What is this GHKL? And that's actually just a name from a founding member of the ATPase family, gyrase, H-protein 90, histidine kinase, and mut-L. That's why we call it GHKL. Now, this is the ATPase activity assay using recombinant CRT1 protein that is expressed in E. coli. And here we use ATP and GTP. I believe that's gamma P32. Here's a CRT1, and CRT1 that has a deletion in the ATPase domain. And here we have commercially available ATPase. And finally, SABP2, that's not relevant protein that we thought that not going to have any ATPase activity. So this is serving negative control, this is the positive control. When we did that, we found that CRT1 has not strikingly high ATPase activity, but nonetheless there is a consistent activity being detected. And if we boil it, we were able to kill that activity as well. And it's only utilizing ATP, not the GTP. So we proved that the CRT1 can be ATP. It turns out the CRT1 has homologs. In fact, 6 additional homologs in Arabidopsis. And the red arrow indicates the Arabidopsis homolog members. Arabidopsis homolog members. Here we have CRT1 and CRTH1, homolog 1, homolog 2. And there's additional branch. And we now have 3 clades. And these seems to be the ones that involve in plant immune responses. What is also known is this CRT1, CRH1, H2 are highly homologous, having about 80 to 90% amino acid identity. Okay? So they actually functionally redundant as well. And they are actually located right next to each other. So we suspect that this gene is tandemly triplicated at some point. So this is one obvious experiment that we wanted to do, assessing the resistance against the TCV, because that's where the genetic screening was performed. And here we have Arabidopsis fully grown the way we infect the plant was we infiltrate the infected plants 2 to 3 leaves. When the Arabidopsis become about 3 to 4 weeks old, and then we assess the resistance symptom in about 3 to 7 days. And sometimes we also look at the systemic portion of the plants, systemic leaf, and also look at the inflorescence as well. So here we can see the Columbia 0, that's another ecotype that is susceptible to Tony-Kunkel virus. And DI-17, as I indicated earlier, because it has HLT, it is a resistant ecotype. And it turns out DI-17, actually you can clearly see there is a tiny little spot that is a hypersensitive response that is due to the resistant response. And if you let it go a little longer, you can clearly see the Columbia now beginning to develop a Callose. So here's inoculated leaf, here's a systemic leaf. And DI-17 still have very discrete portion of cell death, hypersensitive response, but that still seems to be the same. But when we examine the CRT1 mutant, there seems to be some Callose being developed. Okay, and then we realized that, hey, this one already have 2 almost identical genes sitting next to each other. So what we tried to do was, why don't we just silence those 2 additional members? And here we have HBCRT1. This is the mutant CRT1 mutant background, and we deliver the silencing construct that targets CRH1 and 2. When we did that, we found that this disease symptom was much more profound compared to mutant background, and more so as compared to DI-17. And we now begin to see that there is a systemic portion of leaf begin to show some disease symptom as well. So next approach was, let's try doing this one in quantitative fashions. So what we have done here is Western blot using the anti-CP antibody. So this antibody will recognize Tony-Kunkel virus called proteins. And we made a 4-time dilution series. Every time this is 10-fold dilutions. And I just want you to draw attention to Columbia 0 versus DI-17. You can clearly see even in 3 days after infection, there are much lower amount of TCV being accumulated. But this difference is much more greater if you're looking at 8 days after infection, particularly if you're looking at the systemic portion of the leaf. We rarely see DI-17 carrying this TCV, suggesting that DI-17 rarely allow the systemic spread of the TCV. That's the indication of the resistance of HLT. We examine the CRT1. Now this one beginning to allow the systemic spread of the TCV. When we examine this mutant plus silencing transgenic, we found that this TCV is propagated much higher. That indicates CRT1 and its family member is involved in HLT-mediated resistance. So that was the conclusion we draw. And this is actually taken about 3 weeks later. And this shows DI-17 setting nicely. The seed is being set very nicely. And Columbia 0 is setting little or no seeds. That's a typical symptom. And when we examine CRT1 and CRT1N line, we saw they are suffering pretty much. Using the TCV system, we move on to the Columbia 0 now. And if you eliminate HLT, as you can remember, that's a stage number 1. That's a basal resistance. Now we're not talking about ETI, we're talking about basal resistance. So we are using Columbia 0 background. Here's a wild type. And we utilize tDNA mutant that is available through the data stock centers. That's something that I was trying to remember. Okay, so we obtained this tDNA mutant from the stock center for CRT1, CRH1, and CRH2. And later we realized that CRH2, if you knock it out, it becomes lethal. So we were not able to use it. So today I'm only going to show CRT1 and CRH1. What we found was this is about 3 days after infections, and you can clearly see even within 3 days, this double knockout that's knocking out CRT1 and CRH1 beginning to show chlorosis. And once again, we have done quantitative analysis. Here we have wild type versus a double knockout. This is about 1.5 days after infections, and we begin to see that the CP, coat protein, being accumulated, and that difference becoming larger and larger. Although, since wild type is susceptible ecotype, you expect that the TCV is being accumulated as well. But as compared to wild type, we were able to see the resistance to TCV in Columbia zero background was compromised. So that conclude that the CRT1 not only involved in ETI, but also involved in basal resistance. So next question we had was, okay, so far we have tested the virus. How about bacterial pathogens? So we use Pseudomonas carrying ABRPT2. This is something that I have already introduced. RPS2 is corresponding resistant proteins. So that's the one that we recognize. Here we have wild type and RPS2. This is the control. RPS2 is the mutant that doesn't have RPS2. Therefore, this one completely lost ETI2, Pseudomonas syringae carrying ABRPT2. And we measured the resistance by just measuring the growth. Please note this is the log scale. So wild type only allow about 4 to 5-fold increase of bacterial pathogen. RPS2 is minimum 1,000-fold more compared to wild type. And when we measured CRH1, CRT1, and this double knockout, we saw gradual loss of this RPS2 mediated resistance. So that indicates CRT1 is involved in antiviral resistance and antibacterial resistance. So now we're expanding the story. Another one that we tested here is Columbia immunized with FLAG22. FLAG22 is active epitope of flagellin. So you don't have to infect the plant with a full-length flagellin. You can just infiltrate a FLAG22, that's 22 amino acid long peptide. You just infiltrate it, and they can trigger PTI, PAMP-triggered immunity. So here we immunize with FLAG22, same thing. We measure the growth of the bacterial pathogens. MORC is control and FLAG22 immunized. Generally, we saw about 10-fold difference. When we tested this double knockout, we saw about 4 to 5-fold difference. Yes, it is a very small difference, but we were able to see a very consistent difference every time we test wild type versus double knockout. And FLS2 is the one that doesn't have PAM recognition receptor, so therefore you don't see this difference at all. So that indicates CRT1 also involved in PTI. All right, so this is another way you can demonstrate PTI is compromised. You are measuring reactive oxygen. This is luminescence base. You actually measure luminescence, and that will be a reflection of reactive oxygens. You measure it for about an hour. Here's a wild type treated with the FLAG22. You can see that there is reactive oxygen productions, and that production was quite compromised in the double knockout mutiny. We also analyzed Callose deposition in response to flagellin. Here's a wild type Callose being deposited in 16 hours after infection. This is aniline blue staining. So you're actually looking at Callose that's modifying the cell wall, is fortifying the cell wall so that the patient will have a hard time to coming in. So here we have Callose being deposited in a wild type. FLS2 doesn't have such a response, and double knockout actually shows the pattern closer to FLS2. Another one, if you trigger immune response all the time, what plant suffer is growth, because immune response is energy consuming process. So you don't want to turn on all the time. So in this case, you treat the plants with the FLAG22 in the wild type background versus the FLS2. You can see that there is a difference in growth. Here we're measuring root growth, and the FLS2 is growing like as if it doesn't see any FLAG22. And when we measure against this guy with the double knockout, we were able to see that yes, the growth retardation occur, but not as significant as wild type. So altogether, it suggests that CRT1 involving PTI. So summary one, this is take-home message number one, okay? So here we have CRT1, I identified through map-based cloning, and we found that it has ATPase activity, and that ATPase belongs to GHKL superfamily. There are a number of slides that I didn't show due to the time limit. This is what we have found. So in addition to ETI, PTI, basal resistance, we also found that non-host resistance, systemic acquired resistance is also compromised. And we also tested in the barley, tomato, and potato. They all have immune functions. The CRT1 homolog in barley, tomato, potato, they all have similar functions. We are in the process of publishing that manuscript right now. Now, here's another selling point. So CRT1 is involving in the wide range of immune response in plant. And because the nature of screening, we thought that perhaps this can interact with resistant proteins. Okay. The way that we try to prove those possibility was we utilized nicotinamide bentamiana, co-expressed these two components, CRT1 tagged with a MYC and arginine tagged with HA. And you express a couple of days, so then this nicotinamide bentamiana leaf will be full of proteins. And then what we have done is pull down with antibody that targets one of them. So in this case, we targeted resistant proteins. So we'll pull it down, ask whether or not CRT1 co-immunoprecipitated together. So I want you to draw attention to the top panel here. So we're analyzing minimum, I guess we're testing eight resistant proteins. That maybe was good news or bad news because they all interact, except the GAPHA, which is serving as negative control. Okay. So here we show that a wide range of our proteins interacting with the CRT1. The first response from my colleague was, hey, that gotta be flawed. Okay. So here's another experiment that I've done. Here's our protein. If you overexpress our protein in heterologous systems such as nicotinamide bentamiana, sometimes it will trigger autonomous cell death. It will trigger just cell death without any reason. Okay. So we overexpress RCR1, that's from again Arabidopsis. RCR1 is another antiviral resistant proteins in Arabidopsis, recognizing cucumber mosaic virus. And if you overexpress it, unlike HLT, it triggers cell death. Here, panel A is just showing a regular leaf, and if you bleach this nicotinamide bentamiana leaf, the cell death is better shown. This is bleached with ethanol. You can see that there are brown spots. That is an indication of the cell death. There is a couple of mutants in RCR1 that blocks the cell death. So here we utilized two different point mutant. And then we did a co-IP. So here we can see that RCR1 did not interact with the CRT1. However, when we use the mutations that abolished autonomous cell death, now begin to interact. Okay. So I introduce another scenario. Okay. How about if I co-expressed our protein together with the CRT1? But in this case, why don't I just introduce effector to activate those resistant proteins? So that's what I have done here. Is I co-express CRT1, HLT1, and then introduces TCV or mutant TCV. The difference between these two is this mutant TCV is not being recognized by HLT. So it's not going to trigger cell death. You can clearly see here we have TCV triggering the cell death by activating HLT. And I just followed it 2 days, 2.5 days, and 3 days after infiltration. The interaction was diminished over time, indicating that activation of our proteins somehow triggered the dissociation of CRT1 from our proteins. So the interaction between resistant proteins and CRT1 can be dynamic. It doesn't have to be associated all the time. Okay. Now, Here we try another immune component that is FLS2. This is the first, the component that I introduced in the first slide. This is the PAMP recognition receptor. So it will recognize flagellin, and it will trigger the signal downstream. Here, what we have done here is we generated a transgenic line in Arabidopsis. So previous experiment was all done with the Nicotiana pentamiana. Now we are moving on to homologous system. Arabidopsis expressing CRT1, this is genomic clone using own promoter, own terminator, and just tag with a MIG in between. Okay. And what we have done is this is input. You can clearly see in the wild type you don't see any. Only can find it. The CRT1 is found in only from the transgenic line. And FLS2, we just use antibody detect FLS2. We got that from Andrew Bent's laboratory from University of Wisconsin. It is very weak. IP, we pull down with a mix, so now we're pulling down the CRT down. Ask whether FLS2 coming down together. The result was, yes, there is a very tiny band, but it's very consistently interacting. We're not saying this is a really strong interaction, but there is a consistent interaction between CRT1 and FLS2. A lot of times, if you find a mutant that is compromised in immunity in plant, it's sometimes due to the fact that the immune protein is not stable. I think HSP90 is one of the cases. So here we ask whether or not FLS2 is unstable in the double knockout background. Is it the case? So here we're asking such a questions. Here's a wild type, and here's a double knockout. You can see that the amount of FLS2 being detected is comparable. So we claim that the stability is not being changed. We also have done the experiment to show RPS2 protein, that's a resistant protein. That protein stability also wasn't changed. So we don't think that stability is anything to do with the CRT1. This co-IP experiment is showing the FLS2 interaction with the CRT1 can be dynamic. So in our protein interaction and CRT1 interactions, if you activate the resistant protein, the interaction falls apart, right? So here we try to activate the FLS2 by adding FLAG22. And we're trying to see the interaction between the FLS2 and CRT1. And make the long story very short, we didn't see the difference. So this is the confocal analysis looking at the localization of CRT tagged with the GAP. This one's supposed to be a T series. Unfortunately, the file lost. So you have to believe me what I'm going to say. Here's a GAP signals, and this is I think the peroxisome marker. And I was trying to co-localize them, but they never do. But they tend to move around. They have very rapid cytoplasmic streaming. We do not know what the significance is it, but whenever we're trying to look at under the microscope, either using the Nicotiana pentamiana or Arabidopsis, we see the same pattern. So they follow cytoplasmic streaming. They localize to punctate the vesicles. That's what we know. We claim that it is part of the endomembrane system, but we do not know where exactly it's being ended up because we tested peroxisome and many other markers, including the early endosome markers. We couldn't see any co-localizing marker so far. But around the time we finished publishing this, we found that the CRT1 can go to also the nucleus. So here's what we have done. So we utilize the same Arabidopsis transgenic line expressing genomic clone of CRT1, the same line where I have used co-IP experiment to show FLS2 interactions. Here we're using it in the TEM, transmission electron microscope. So here's the zoomed out portion, and this box just show where that is being highlighted, and this is just mark treated. And we found that there is an occasional localization of CRT1. This is the immunogold indirect labeling. And what we have also found was if we treat it with the FLAG22, this number of CRT1 locations increase, but also they also tends to aggregate together. You can see that they are sort of individually localized, but you can see that these CRT1 localized aggregated together. Okay. So this just showed quantitation of several different images of TEM, just show that the CRT1 goes into the nucleus. This one backed up by the biochemical purifications. Here we are doing nucleus enrichment, and that enrichment was further confirmed by this two antibody, PEPC and anti-histone antibodies, to ensure this nuclear fractionation is okay. And then we ask whether or not CRT1 is present in the nucleus enriched proteins. But you can clearly see even after the enrichment, the amount of the CRT1 is very, very low. So the portion that goes into the nucleus is very, very small. That's the reason why I was not able to see that when I use a confocal. So, but also we also found that in the presence of FLAG22, we saw that that is the amount, although it's not as significant as I want it to be, but it seems to show a consistent increase. We tested again with now Pseudomonas, 3 different Pseudomonas. This is the HARC-C mutant that doesn't have functional type 3 secretion system. So this is essentially flagellin. Okay? And then PST is a virulent pathogen. Here is avirulent pathogens. This is 16 hours after infection. So there's a time difference. This is the 10 minutes after, and this is the 16 hours after. And when we assess the amount of CRT1, we particularly see enhancement of CRT1 in the nucleus when we infiltrate with avirulent pathogens. So ETI seems to further enhance localization of CRT1 into the nucleus. So here's the tryptophan fluorescent analysis. The point that we're trying to make here is this tryptophan fluorescent unit can change if the protein change the conformations. Here we added DNA and RNA. Essentially, we're trying to ask whether CRT1 is DNA interacting protein or nucleotide interacting protein. And hoping that perhaps the TCV has a higher affinity as compared to other DNA. We put in the TCV DNA, TCV RNA, Easter RNA, lambda DNA, whatever available in the laboratory, and then we try to see the difference. But what we have found was there is always interactions if you put any nucleotide. Looks like it's a non-specific fashion. And here we have, if we denature the CRT1, then this DNA nucleotide interaction is abolished. So this is activity-based. So now we have CRT1 can go into the nucleus, and it can interact with the nucleotide. So how about we mix any DNA and see is conformation change occur in the DNA template. So here we mix with just regular plasmid, any plasmid you can find in your refrigerator, and then you mix it with recombinant CRT1 with a different divalent ion, because a lot of nuclease requires different divalent ions. So manganese, magnesium, zinc has been tested. Here is a positive control. This is the nicase from Nicotiana bentamiana. You can see that generally if you run just a gel in the DNA in the agarose, this is supercoil, they migrate faster. Once it becomes relaxed, so if you have a nick, it will just release all the supercoil, so it will migrate slower. So you can see the two bands. And if you mix with the nicase, this supercoil DNA become all relaxed. So you can see that the upper band is becoming predominantly higher. And we found the CRT1 as you increase the CRT1, this activity going, this amount of relaxed DNA goes up. And if you delete the portion of portion that we think that is responsible for endonuclease activity, then that endonuclease or nuclease activity is gone. Okay. So we think that this CRT1 at least possesses manganese-dependent endonuclease / nuclease. The significance of it, we don't know yet. Okay. Another thing we have tested is, yes, okay, so if the DNA is interacting with CRT1, and CRT1 can be present in the nucleus, how about we testing the activity or capability of repair or recombine the DNA? What you can do is if you subject this plant, Arabidopsis plant, with a DNA damaging agent like mitomycin C, even with wild type, as you increase this DNA damaging agent, the plant percentage of plants shows true leaf decreases. Somewhat dramatically, 80 to 120, you can see that now majority of the plant doesn't develop true leaves. Okay? Now, SSN1 is from Jin and Dong's laboratory. This is suppressor of SNE1. They show that it is hypersensitive to DNA damaging agents. So this is serving as control. So we subject that treatment with double knockout. We found that it is actually tolerant to these DNA damaging agents. Okay. So by knocking out, it's becoming tolerant to DNA damage. That's all the clue, but we don't know exactly why that is showing this. So this is what we have reported so far. So take-home message number 2. So I showed you the CRT1 interact with diverse immune-related proteins, R protein, PLR protein. We also shown that the back one, big one, they all interact with the CRT1. I believe to date, this is the only protein that is shown to interact with both R protein and PLR proteins. So that's a good selling point, although one could argue, hey, that looks non-specific. But I showed you that the CRT interaction can be also dynamic. And CRT1 family function only algin-mediated resistance. This is something that I haven't shown you. Upstream of MAP kinase cascade and calcium signaling, we reported that as well, but I didn't show it today. I've shown you the dynamic interaction with these two R protein and PRLs. And endomembrane vesicles seems to be predominant place. But now we also found that found CRT1 in the nucleus as well. Okay? And it looks like CRT1 accumulates, although it's a very marginal amount as compared to all the pool that is available outside of the nucleus. It seems to be accumulating in the nucleus upon its activations. And it looks like CRT confers susceptibility to DNA damaging agent. Remember, I was using the knockout, so therefore I conclude that CRT1 in the wild type form is conferring susceptibility to DNA damaging agent. Now, this is the final section of my talk. This is just emerging area of plant immune research right now, and a lot of people now beginning to find that epigenetic components or chromatin alteration play another role in plant immunity. Okay. So remember, the section number 1, section number 2, what I have told you. Section number 1, CRT1 functioning wide range of plant immunity. Section number 2, I showed you they are interacting with large number of plant immune components. The unique thing about this CRT1 protein is a lot of immune associated protein that show the PL gene or this marker being altered. But if you examine the CRT1, it doesn't show. So that's very unique. And that's something that I had a hard time explaining. I don't have PL1 being changed, but they are saying that has to be changed. But now, we think that there is another clue in plant immune responses. That's what I'm going to show you. And the epigenetic and chromatin alterations, there are a number of components that is being shown. I think that include AGO1 and other component is shown to be important in plant responses, but we do not know how that works. And last year, Steve Jacobson at UCLA published science paper, and what he has done, his group has done here is they screened the mutant, restored GFP silencing, and they found that two mutants, CRH6 and CRT1. That was very confusing at the beginning. Wow, that one has a gene silencing function too, but how? And this is another eye-catching figures from our end. You can see that here's a CRT1 CRH that is tagged with a MIG, and this is immunofluorescent data. And here's a DAPI. So DAPI will stain the nucleus, and this is the chromocenter. Chromocenter is a part of the chromatin, and generally stain heterochromatin area. And when they combine these two, they found that this CRT1 goes into predominantly heterochromatin area based on chromocenter locations. Okay. So what we have decided to do, how about double knockout mutant we have used? By the way, there is two additional publication made showing that CRT1 and CRH6 is involved in RNA-dependent DNA methylations. So it is showing more and more functioning in epigenetic area. So summary number 3, take-home message. So CRT1 family may be involved, a lot of may. Heterochromatic formation, histone compaction in a heterochromatic area, and maybe modulating histone methylations. The title was molecular clue. I'm still trying to gather clues. So I'm hopeful that if I have another chance to coming in here, I should be able to give you a better picture next time. So that's about it. And this is the people that this project possible. I have one. Here is Yogi Boria, who is a very talented PhD student. Section 3, I think most of the figure was generated from Yogi. And Jichul Nam is another PhD student in my laboratory, just joined. He's been about two months. Rachel is another undergrad who's doing another part of my project. And Yuna Kim is a technician in my laboratory. Chad Mass is a high school student, sometimes work in my laboratory, sometimes just go anywhere where I don't know. I had one very helpful postdoc for about a little over 1 year, who's now in Ping's laboratory, who's in my audience right now. And here's two groups that I have collaborated from Germany and Boyce Thompson Institute. I'm still collaborating with Dr. Classic. And these are the people that I have collaborated over the course of this project. Thank you very much. I'm ready for your questions. Yes.
I guess I'm still wrapping around the relationship on the pump. How can the CRT1 is all in the first slide you showed that it was localized in the recipe of the mostly, right? But it's also interacting with the R proteins. It's acting somehow sequence to them or something, before they're activated and interact with the effector protein. But it also goes to the nucleus. And then also functioning in compaction of the histone. Is this function related, or are they distinct? And how is this localization affecting function?
Here's my hypothesis. Okay, I didn't connect it because I don't want to over-interpret my result. But my hypothesis is, now let's start with endosome localizations. It is vastly localized in the endosome. And we know a lot of our protein also localize the endosome as well. Okay? And then once this R protein is being activated, I believe, you remember also I showed you that CRT1 increase, the level increase upon activation. So something has to come from the vesicles. And I hope that whatever comes into the nucleus is the one that has been activated through activation of R protein, through activation of FLS2. Now, once you get into the nucleus, what it does, perhaps perhaps CRT1 is the one that negatively modulate genome stability. It's been known that under biotic stress, genome stability is going to be down. And if we need to balance, okay, under biotic stress, the genome stability will suffer. And that probably can be utilized in evolution over the long run. But you want to also make sure that you want to double check this stability is not going out of control. So I think the CRT1 can be a negative modulator. But then why then, when you lose CRT1, is losing immunity? Here's my overly oversimplified hypothesis. If you lose CRT1, that means genome can be unstable even before stability, the biotic stress. So there will be slightly elevated stress response already. So the cell going to be habituated, somewhat desensitized. So when you challenge with the pathogen, that immune response is not going to be as robust as it should be. Because of that, when you lose CRT1 or its family member, the immune response is weaker. I actually went back and asked, is the PL gene is somehow induced in the basal, the cell that hasn't seen the pathogen? That was the case. So I have one or two supporting evidence, but I had to do a lot more to support that my hypothesis. That's the reason why I didn't share that today. But anyway, since you asked it, I'm sharing it. Yeah, right. Yes, Mike.
So why not to, since CRT you believe affects the ability of heterochromatin areas, that should be reflected in expression pattern, in a change of expression pattern. So let's say more expression, or rather less expression in the mutant, less expression of defense genes. Why for your analysis you didn't pick up gene defense genes? You pick kind of random, you pick copia and something else. Why not to directly look into defense genes and see if they're altered in expression pattern? Or just now, RNA-seq analysis on CRT?
You mean in double knockout mutant context? I have done it already, and I've analyzed the transcriptome of double knockout in responding to Pseudomonas syringae. Back then, I think RNA-seq wasn't popular. It was DNA chip, RNA chip analysis. At that context, I couldn't see much PL being elevated. So that's why I ruled that out back then. But now I'm actually going back and check and basal, because I only look at the induced one and see the change. The change was very comparable. So and there was not statistically significant. When I looked at the basal now, they look different. So that's where my argument was coming from. But I looked at the PL1, PL2, PL5 at the beginning. And I reported only that there isn't any much difference. Yeah.
So what is the phenotype of CRT1 and CRH6?
Phenotype.
Yeah, that was published by the PCLA group. And why didn't you add it as your control to the double and triple mutants? Why didn't you add it to CRT1 and CRH6?
Okay, so the first question, do I see the phenotype of any CRT1 single, double, triple mutant? Double knockout, if someone give me double knockout and wild type, I can distinguish. Very subtle morphological phenotype. Little curly. But other than that, it looks fine. CRH6, if I add CRH6 making triple knockout, they don't look different either. If you go to the single knockout, they are wild type. If you mix it, I cannot tell. But double, triple, I can tell. That's a morphological phenotype. Why did I add the CRH6 a little later? Because I learned that last years. So we began processing it starting last year. Now all the experiments that we do it in the lab now include double, triple.
And to follow up the question, so that paper that you published in Science, did they find out some of the function related to the CRT1 that you so far you were summarized out?
I hope not. I hope not. I graduated from UCLA, and I know Steve Jacobson very well. And we are sort of good friends too. I'm hoping that we are not stepping on the toe each other. I'm only focusing on the plain immune side. And what they have reported in the science is it has a function in gene silencing and localization, and that's pretty much it. And nothing beyond that point. I'm sure he's also working on the follow-up study, but I don't know. I don't have the information yet.
My final question. You see double bands in your CRT or two.
Okay.
That, I
Frequently asked questions. I should have explained it. So whenever we express it, CRT1 forms a doublet. The upper one is full length. The lower one is truncated form. And we know where that is being truncated. And the function of it, we don't know yet. But we do know the upper one is full length, and the lower one is the one that has C-terminus being processed. And we do know that the lower one is the one that losing the interaction rapidly.
Could they be forming a dimer,
Oh, dimerizations. We do know that CRT1 dimerized. And we have done the H2 analysis. The clone that we obtained was about 14s. That include the PINGs, the CPL3, we already know. And also, it also fish out the CRH6. At that moment, we knew that CRT1 family member heterodimerized it, and we followed that up and found the CRT1 homodimerized, CRT1, CRH6 heterodimerized. And I think it's intriguing to see that so far all this screening performed only identifies CRT1 and CRH6. Yes, yeah.
Can we have some insight from CRT2, 3, 4, 5, 6?
Oh, CR2456. Okay. Here's something that I have to lose when I take CRT1 project from Dr. Classic. What we have decided to do, I'll be working on CRT1 Arabidopsis project, and any part of Arabidopsis. I give up the CRT2, 3, 4, 5. CRT2 has been roughly mapped to chromosome 2, where no immune mutant has been identified. So that has to be novel too. And one postdoc in his laboratory, Dr. Classic laboratory, is working on it. So we should be able to hear that news fairly soon. 3, 4, 5, I think it's very unstable one, so we gave it up. Yeah.
Thank you very much.
Okay.
We're going to continue the discussion at approximately 9, is that right? We have a good day.